Just welcoming everybody to the conference. Many of us are avid and enthusiastic users of the NHGRI catalog, and today's activities, the talks from different speakers, will highlight different ways of using it, different ways that people are thinking about using the catalog for testing hypotheses and trying to learn more about common diseases and complex human traits. We might as well just get started. And of course, the thing I don't have is the agenda, but I think the first speaker is Lucia, right?
Correct.
Helen?
Thank you, Nancy. I'd like to add my welcome to this webinar focusing on current uses and future directions for the NHGRI GWAS catalog. My name is Lucia Hindorff, and I lead the GWAS catalog effort for NHGRI, where I'm an epidemiologist and program director in the Division of Genomic Medicine. Could I just confirm with Nancy, can you see the slides on the WebEx, Nancy and Helen?
I can.
Okay, great. Thank you. So I will be starting by giving an overview of my talk today, which will begin with a summary of the goals and logistics for the webinar. I'll then describe how the catalog handles curation and data display, as well as some recent enhancements we've implemented. Because the central focus of today's webinar is how the scientific community uses the GWAS catalog data, I'll briefly describe a few examples of community adoption and uptake, leaving much of this to the rest of today's speakers. Finally, we'll leave some time for questions and comments from you, the audience. Okay, so let's start with the goals for today. For this webinar, we had a small organizing committee comprised of myself, Helen Parkinson from the European Bioinformatics Institute, or EBI, and Nancy Cox. We've put together an agenda to showcase many of the uses of the catalog. As you follow along, please keep in mind the following goals of the webinar. First, to describe the overall goals of the GWAS catalog. This will be primarily in my talk, although you hear some themes in other talks perhaps. The evolution of the GWAS catalog will be next, including the history of how it originated, the process by which we curate data, and improvements. This will be covered in my talk as well as a little bit in Helen Parkinson's talk later on. And we have a diverse, exciting set of speakers who will help accomplish the next three goals, which are to assess the community use of the catalog and the challenges in its use, to identify possible future uses and needs, and to obtain advice on future directions for the catalog. We hope that the audience will contribute to these discussions as well. Notably, after this webinar today, we will do a video archive and continue to accept questions after the webinar as well. So as I mentioned, we have an exciting lineup of speakers with a broad range of expertise. Following my talk today will be Michael Clark from the University of Queensland, Marilyn Ritchie from Pennsylvania State University, John Stam from the University of Washington, Luke Ward from Massachusetts Institute of Technology, Karen Svensson from the Jackson Laboratory, Nancy Cox from the University of Chicago, Helen Parkinson from the EBI, and Benjamin Neal from the Broad Institute. And we'll end with an open discussion where we can continue to receive feedback and suggestions on the catalog. So I'm going to cover a little bit of logistics here. You'll find everything you need at the web page shown below, including dial information, the agenda and speaker biographies, and a link to the video archive when it becomes available. So chances are that if you're watching this, you have this information already, but if you're watching it live and need to come back later, you'll find everything you need on this page. So a few additional logistics notes. Presenters are joining us today online and over the phone from literally all over the world. Some people have even made time to give their talks while they're traveling and away from their offices. So please keep in mind that we'll do our best to keep things running smoothly, but we are at the mercy of the wonders of modern technology. So please bear with us if we happen to come across any glitches. We will also adhere to the agenda times as listed on the website, which means that if a talk ends early or if there are fewer questions than we had planned for, you may hear silence until it's time for the next talk. We have allotted time for questions directly after each talk. This is the time to ask them because not all presenters will be able to attend the entire webinar due to time differences. Those joining us in real time can dial *1 to connect to an operator to ask questions or use the chat feature in the WebEx window. As a last resort, or if you are joining us through the video archive, you can send an email to the address you see here. Okay, so with those details out of the way, let's go ahead and introduce you to the basis of the GWAS catalog curation and display. So 8 years ago, a GWAS on age-related macular degeneration was conducted, identifying a signal in the complement factor H gene on chromosome 1. Since then, a flurry of activity has culminated in over 10,500 variants associated with hundreds of traits and diseases in over 1,500 peer-reviewed publications. Now, most of you will recognize that these data come from the GWAS catalog, which systematically curates and disseminates published GWAS data in an online, user-friendly, downloadable resource at the top URL that you see here at the bottom of the screen. When this resource was initially established, the idea was to define a framework with pre-specified criteria for inclusion, to systematically and regularly curate data from published studies, and then to disseminate it publicly. One important way in which we do that is to continually update this diagram and make it available as a visual summary, which many of you will be familiar with. Shown under that URL is the URL for this diagram, which has now been automated and which I'll touch upon further later in my talk. You may be interested to know a bit of the history of the GWAS catalog, which had its humble origins as a series of word tables in a paper authored by Terry Manolio, Lisa Brooks, and Francis Collins. Those tables summarized what was known as a few dozen GWAS papers available at that time. Those of you familiar with the catalog will recognize the structure of the table and its headings, which include disease trait, sample size information, and then SNP disease association information. This has carried over into the structure of the GWAS catalog page in its current form and the data file that you can download for your own use. As that paper was going to press, we released the catalog publicly on our genome.gov page in March 2008. Since then, we've continued to develop the catalog, resulting in the 1,000th SNP curated and a descriptive paper published in PNAS in 2009. 2010 saw the curation of our 500th paper. In 2011, we curated our 5,000th SNP and 1,000th paper. In 2012, our collaborators at the EBI automated the GWAS diagram, and earlier this year, we curated our 1,500th paper and 10,000th SNP. Truly, the GWAS enterprise has been a prolific one. Many of you are familiar with the catalog homepage, which is shown at the bottom for the URL. Note that we offer several options near the top of the page for users to view the interactive diagram, which is on the EBI website, to view and browse the full catalog in HTML, to download the catalog in a text file, which you can work with further, and to search the catalog based on several parameters. You'll also find links to the descriptive paper, more details about our methods, and other resources elsewhere on that page. So the search screen enables searching by journal, first author, disease trait, pre-specified chromosomal region, gene or SNP, odds ratio threshold for diseases which are binary, and p-value threshold. This is partly our way for helping people find results that they may know that they're interested in based on criteria such as author or gene or SNP. So once you search for a study, each study is summarized in a series of HTML rows. Study level information summarizing the citation, disease trait, and initial and replication sample sizes are shown. I had to split this into two since it's a little bit wide. The other half of the set of rows shows SNP level information, including the genes reported by the authors in the paper, the gene or genes mapping to that location in the genome, the RS number and risk allele, genomic context, risk allele frequency, p-value, and odds ratio or beta coefficient. We also include some basic platform information, phenotyping platform, For ease of display, only a limited number of SNP disease associations are shown. Clicking on this link that you see here displays the full set of SNPs extracted from this study. In terms of curation, this workflow diagram summarizes our process for curating data and improving the catalog. First, we identify potential studies using a weekly PubMed literature search, in addition to a daily news clipping service. Studies must meet eligibility criteria as summarized on our webpage, the most important of which is attempting to at least assay 100,000 genome-wide SNPs before quality control is attempted. Curation, which is done in concert with the EBI, consists of our curation team thoroughly reviewing each paper, including supplemental materials for eligible SNP disease associations. Again, these are described more thoroughly on our webpage, but they include associations for which the p-value is less than 10 to the minus 5. We have a system by which both study level and SNP disease level information is extracted and then double-checked by a second curator. The information is then sent for a quick quality control check and additional annotation provided by NCBI before it is published online. Our collaborators at EBI produce the automated and interactive diagram. In the near future, we are working on systematically extracting ethnicity information, as well as taking advantage of expertise in curation, informatics, and ontologies at the EBI and data mining at the ISI at the University of Southern California. Before I wrap up this section on curation, I'd just like to give you a peek at our data entry system, which we do completely online. As you can see, study information is populated mainly manually. We also have a separate screen for entry of SNP level information, which again is primarily done manually. We hope to take advantage of opportunities to automate some of this data entry, as curation time is our primary bottleneck in terms of getting data into the catalog. So, having just summarized our curation and display process, I'll now move on to sharing some recent enhancements. As we observed increasing use of the catalog and received input from the scientific community, we began to think about targeted improvements to the catalog. At the top of the list was automating the generation of the GWAS karyogram that you saw earlier, which was initially generated through a painstaking process of working with a medical illustrator to manually place new GWAS findings on the karyogram every quarter. Automating the diagram would not only reduce the effort with updating it over time, but would allow us the ability to make it interactive. Working through a collaboration with the EBI, we implemented this feature as well as a few others. Using an ontology, which we'll hear more about later, we consolidated the number of traits into higher-level categories, resulting in fewer colors and a simpler display. A dynamic time series display is now implemented, as well as interactive filtering on traits for select browsers. So the automated diagram is at the link that you see here at the top of the page. I'll just take a moment to briefly walk you through it. You can see that it displays, and the interface is similar to Google Maps, so you can drag the diagram around and, more importantly, use the scroll bar on the side there to zoom in and out. So if you hover over a dot, as shown with this red arrow, there will be a pop-up box that gives you some more information about that SNP disease association, including links to Ensemble SNP, the experimental factor ontology mapping, which is the ontology that we use, the study citation, and the GWAS catalog record. And note that as you zoom in, you can kind of highlight that region of the genome closer and closer without much loss of visual clarity. We also have at the top of the page the ability to browse trait-specific views. This is an example of a GWAS catalog diagram filtered on type 2 diabetes, so only those dots are showing up. Here in the middle, I've shown you the pre-canned diseases for which we've generated filtered views, and these were based on the most popular searches for the GWAS catalog. You can also browse the time series views and scroll through them. All of the image files that you see, as well as the ontology files, are available on the Downloads tab for you to be able to use for your own presentations or for your own scientific efforts. Additionally, we have a Help tab which describes some basic features of the website and may help you navigate some of the website a little bit better. And then the About tab includes some documentation. In the near future, we hope to implement additional diagram improvements, including interactive links to other genome browsers, improved filtering features. We'd like to sync this process up of generating the automated diagram with the curation process so that we can make updated data available more frequently and possibly in real time. We're working on improved browser compatibility, and then, of course, we'd love to hear your suggestions. I'd like to switch gears a little bit and tell you an additional area in which we've tried to improve our curation, which is for information related to ethnicity or ancestry of study participants, as reported in GWAS publications. So many of you are familiar that in the United States, we observe disproportionately high disease burdens in non-European populations. Studies that have looked at the frequency of variants identified from the GWAS literature have observed variation of up to 40-fold between populations of different ancestries. In addition, in 2009, a study was done and looked at all of the studies in the GWAS catalog and observed that the studies were performed at a ratio of 10 studies to 1 for European ancestry versus all groups combined. So taking all of this information together, it suggested to us that the knowledge base generated from GWAS was thus based largely on populations of European ancestry, and we saw a need to at least begin to devote effort to define and extract ancestry information to try and help define this a little bit further. So we developed a framework for extracting ethnicity, and the gist of it is that we capture both structured and free text information. After some deliberation and consulting with some outside individuals, we settled on the following approach. At the top of the box here, you'll see that we have an inferred ethnic and ancestral group that we select for every replication or initial sample size. So this is something that the curators assign. We also read through the methods carefully, and where a country of origin, such as grandparental origin, is assigned by the authors, we note the country. We note the country of recruitment for each subpopulation that's eligible for inclusion in the catalog. And then we also include a free text description. And the intention here is to be able to capture any form of geography that's smaller than a country, as well as ethnic-specific information, such as tribe or information that we feel might be useful to the scientific community. We are currently in the process of backfilling many of our past papers with this ethnicity information, so it's not available publicly yet. But we have done an analysis of 575 GWAS papers from 2011 to 2012, and we observed that 55% of these papers were in European ancestry populations only, 27% were in non-EA populations only, and 13% include those populations. So you can observe here that since the 2009 paper that I referred to earlier, things have improved a little bit, but there's still an overwhelming body of knowledge coming from European ancestry individuals. At the study participant level, 74% of the participants are of European ancestry. And as we'll see in a moment, the countries of recruitment are dominated by those in Western Europe and the United States. So this is a Wordle that shows you the countries of recruitment that are represented in the catalog, and the size of the text is proportional to the frequency with which we observe it. So this makes the point that I was referring to earlier, that the countries are dominated by Western Europe and the United States, although we see Asian countries making an increasing presence. If you just focus in on this little region here, you can see that we do have one or two studies coming from Argentina, Gambia, and Ethiopia, among other countries. But this really does highlight the kind of disproportionate influence that you see coming from certain countries. Okay. Let me show you a Wordle of a different sort to introduce to you how we're improving our trait disease information. So this is a Wordle that is a snapshot of the different trait names present in the catalog, again, proportional to frequency. The size here is not as important as the sheer number of traits that we have in the catalog. There are about a couple hundred of them, on the order of a couple hundred, that form a relatively unstructured trait list. The curators do try to be internally consistent and reuse terms when possible. Although this is more granular, it does not permit grouping of or searching on related terms very easily. So we've worked with Helen Parkinson on mapping GWAS catalog traits to an ontology, which facilitates the broad categorization here. And the basic idea is that by mapping trait terms to an ontology, the visualization of the catalog can be made simpler by categorizing them into a smaller number of traits without losing the underlying granularity of the individual associations, which is still captured at the individual dot level. So you'll hear more from Helen later today about the ontology and how integration of the catalog terms into existing ontologies, such as the experimental factor ontology, will permit systematic grouping of traits and allow more structured and inclusive searches. So let me switch tracks a little bit and tell you a little bit more about the community adoption and uptake of the catalog. The GWAS catalog is enjoying broad exposure across the scientific community, with the catalog data being repurposed for a number of different study types beyond just GWAS. Of course, you'll hear more from today's speakers about how they've looked at GWAS data and incorporated it into their scientific efforts. But let me just give you a highlight of selected findings. For example, the ENCODE consortium utilized the GWAS catalog data to identify regulatory elements that are associated with sequence variants linked to human disease. Another recent example analyzed genes from the GWAS catalog data for which there were drug targets and compared the disease indications of these drugs with the GWAS catalog traits. So several mismatches were highlighted which indicate potential opportunities for drug repositioning. GWAS catalog data have also been used to analyze and draw conclusions about the heterogeneity of genetic associations across major ancestral groups, to make inference about functional copy number variation and protein interactions, and even as a resource of scientific expertise with which to study collaborative scientific processes. So I'm highlighting not any one particular group or effort, but just to give you a sense of the broad range of applications for which the GWAS catalog data have been used. GWAS catalog data have also been integrated into genome browsers that are used by the community, including the UC-SC genome browser, DBSNP and other NCBI resources, and Ensemble. Finally, the catalog has spawned the development of other phenotype-oriented resources, such as the Phenotype-Genotype Integrator, a collaboration with NCBI to integrate GWAS catalog and other GWAS data with the genomic and phenotype-centered resources of NCBI in a browser environment that can be easily browsed and downloaded. The basis for this work was recently published in the European Journal of Human Genetics. So let me lastly highlight some key challenges and opportunities that the catalog team has observed throughout the 5 years or so that we've been curating GWAS data. So the primary bottleneck is still the manual curation. The GWAS catalog is a curated resource, which means we don't accept data from everybody or based on unspecified criteria. We do look carefully at each paper and each table in each paper to make sure that the information that we extract meets the criteria that we set a priori. Automated data mining may facilitate some of these efforts in the future. We've observed that integration with other resources and data types will further biological understanding. Of course, all of you know that GWAS catalog data, and in fact, I think in the giant scheme of things, not every experiment can stand alone, and it's really the integration of data that can really lead us to fundamental biological insights. We've discovered that providing data in a user-friendly way, such as the karyograms, will increase adoption and familiarity with the resource. We try to continue to be receptive to suggestions by the community, and we do hear feedback often from individuals. And then finally, collaborations can enable high-priority improvements. When the GWAS catalog team started, we started with a team of essentially epidemiologists, and so we've really enjoyed collaborating with informaticians and other scientists to help us improve the catalog and other services that we can offer to the community. So finally, I'd like to acknowledge the work of many people that made this catalog effort possible. In terms of the NHGRI, Vivian Bonazzi and Terry Manolio have provided scientific leadership. Peggy Hall and Heather Junkin provide curation effort. Kent Clem is our database guru, and Daryl Leah is the medical illustrator that was instrumental in developing the early GWAS karyograms that many of you are familiar with. We rely on NCBI for some features of our data flow process, and we're thankful to Mike, Jack, and Ming. We are collaborating with the EBI and Helen Parkinson's team, and they've basically done the lion's share of the work on the automated diagrams, so thanks to Tony, John, Simon, James, and Danny for this effort. We work closely with Jackie and Janella on curation as well. So with that, I would like to thank everybody for listening and open it up to any questions that people might have. Thank you.
So, do we all see questions?
I'm looking at the chat window now, and I don't see any specific questions from my talk. I think for individuals who are dialed in, if you want to ask a question, please feel free to dial *1, and that will connect you to an operator.
We could take a couple of questions from the panel if anybody who is dialed into the panel would like to ask a question at this time. And maybe I'll take this opportunity to ask Lucia a question. So, Lucia, right now you only deal with array-based data. Clearly, the technology is changing, and the arrays are kind of biased towards what's on the array, and therefore that does assist with the biasing towards certain populations. We know people now are sequencing populations and starting to do sequencing-type studies. What's your feeling for how important that could be for the future of the catalog?
I think it's actually extremely important to think about sequencing data. So when we started the GWAS catalog, we figured it would have a limited shelf life. GWAS studies were only going to be around for every so often. But the reality is that people are still doing GWAS data. There are more complex arrays that are being developed. And I think the efforts of the NIH and others to deposit the GWAS data and make it widely available have really facilitated the use of those data, which of course continues to be accumulated in publications. So I think GWAS data will continue to be important. We would love to take on sequencing and other array-based data, but curation really is a primary bottleneck. And at the moment, we're doing all we can to keep up with just the GWAS catalog data. But I agree. I think as the technology evolves and we start to look at rare and more functional variation, even at the sequence level, I think those efforts will be important to catalog as well.
I had a question about whether the results of gene-based tests are being included in the catalog.
So our criteria are pretty strict. So if it's a GWAS, if they do sort of a discovery phase that consists of 100,000 or more SNPs, we include it, but we don't include follow-up data. So if for example, somebody identifies a SNP and they follow it up, we don't include that. I think as far as gene-based method analysis.
Gene-based analysis of the individual variant data.
So if it starts off gene-based, we typically don't, because we usually look for at least a sort of a genome-wide, more agnostic type of approach.
Well, but now this would be looking at each gene with a gene-based test, which is, of course, how rare variant tests from, for example, whole exome data will often be reported.
Yeah, I understand it's important. The limitation, I think, of our curation team has not allowed us to expand in those directions. And we did observe when we did our PNAS paper that the vast majority, well, not the vast majority, but a substantial component of the SNPs that we observed were intergenic. So I think about 80% are non-coding. Of course, some of those are in genes as well. But at this time, we actually don't. If it's only a gene-based, we don't include that in the catalog, and I realize that excludes a lot of important work.
Right, because of course many of the gene-based tests now are designed to include non-coding variants as a part of the way a gene is annotated. For example, eQTLs annotated to a gene because they have been associated with transcript levels for that gene. But so it's something to think about going forward, particularly because in order to include the results of studies on rare variants from the sequencing, I don't see how it's possible to avoid gene-based results.
Yeah, I think you've highlighted a very important opportunity for us to think more about. Thank you.
Lucia, this is Marilyn Ritchie. I also had a question. Have you thought about, or are you capturing the SNPs that are being found associated through SNP-SNP interaction models in some of these genome-wide interaction analyses? I know for a while there just wasn't statistical power, but I think we're starting to see more and more of them published that are replicating in other data sets, and I'm wondering if any of those are being captured.
So it depends on the design of the study. So a study design where they're looking at pairwise SNP interactions across the entire genome, or even across the entire genome, by a limited number of SNPs, we actually do screen those for inclusion. So as long as they meet the inclusion criteria, we will include them.
Great.
And that goes actually the same for gene-environment interactions. So if it's a gene-environment interaction study design that does look across the entire genome, we do include that and curate it.
That's great.